Well, hello everybody. My name's Carolyn Leary. I'm chair of the Sheffield ME Fibromyalgia Group, and this would normally be our annual general meeting with a guest speaker, who today is Professor Simon Carding, who I will introduce shortly. We've decided, because of the current circumstances, that we're actually not having our AGM at the moment. We're going to delay that until the autumn, in the hope that more people will be able to attend. So, but we are going to go ahead with having the talk from Professor Carding. So we're delighted that you've been able to join us from somewhere near Norwich. And so Professor Carding, I'm just going to introduce you briefly, and then we're going to run this as a question and answer session. We'd be very happy for anybody to send in any questions as we go through the Q&A, through the interview. And please send those questions in either on the Zoom group chat or on the Facebook Live stream. So we'll be able to pick up those questions, and we'll either feed them in at the end or as we go through, depending on what comes up. So, Professor Carding, you started off with 15 years, I think, of your career spent in the United States, New York School of Medicine, Yale University, University of Pennsylvania, and returned to the UK. We're delighted to have you back, to take up the chair of molecular immunology at the University of Leeds initially, and then 2008 recruited to the University of East Anglia to develop a gut research program, taking up a chair in mucosal immunology at the UEA Medical School, and the position of head of gut biology research program. So the success of this program has led to the establishment of the Gut Microbes and Health Research Program that is now integral to the research agenda at the Quadram Institute. And as I understand it, but I'm hoping you will elaborate significantly on this, what you're particularly looking at is the ways in which gut microbes interrelate with the host and the role they play in establishing gut health. But not only that, but also diseases, not only in the gut, but also in other organs as well. And that's led to some interesting further research into the gut microbiome-brain axis, and the development of another few lines of inquiry. So I'm kind of keen, before we get into the kind of the detail about the relevance to ME, just to sort of, I wonder whether you could start off by just explaining to us more broadly a bit about how the gut microbiome system actually works.
Well, thank you very much, Carrie. So I have in fact prepared a slide presentation which is very much based on the questions you anticipated and your members anticipate asking me. So if I run through the slides first, hopefully that will address most of the questions. And I've sort of I'll try and keep it to around 25-30 minutes at the most, which should hopefully leave time for questions. So if I now share my screen. Okay, I'm just having to make some changes to settings here. Apologies for the delay.
No worries. So we've got a range of people here in the audience, and if you want to text in on Zoom where people are from, that would be great. I recognise quite a lot of names from the Sheffield group, but I think we might have got some people from further afar as well.
Okay.
interesting take on geography is Stockton-on-Tees, just outside Sheffield. So Essex, Somerset, Leeds. Great.
Ah, right. So, can you see my screen, the PowerPoint presentation?
Yes, I can.
Very good. I'm getting thumbs up there from Sean. Good. So I'll just go into full screen mode. Okay. So, as Caroline said, I hold two positions. One is at the Norwich Medical School at the University of East Anglia, but I in fact spend most of my time at the Quadram Institute leading a program on gut microbes and health. So what I've entitled my talk as ME Research at Quadram Institute, Current and Future. So I'll sort of start off by talking a little bit about the gut microbiome and then its relationship to ME and the things that we've actually got planned and are hoping to do when we can eventually return to work. So just by introduction, I'm sure most of you know that ME, the cause of ME are unknown, although there are some significant clues which are coming out from various avenues of investigation, which I've summarized on this slide. This involves immune dysregulation, defects in the neurohormonal connectivity, mitochondrial impairment as it relates to energy, both production and utilization, and more recently, changes in gut microbe populations that may also be a part of this. And of course, viral infections as well have been linked to the trigger of causing ME. Next slide. So the evidence for infection goes back really to the beginning of when a condition that related to ME was first identified in about 1934 in Los Angeles. It was originally misdiagnosed as polio, and then subsequent outbreaks in Iceland at the Royal Free Hospital in London further distinguished it from polio infections until eventually in 1984 it was named chronic fatigue syndrome. And unfortunately this gained the very unhelpful connotation of being yuppie flu. So I guess that sort of led to it not being treated as seriously as it should have been. But I think subsequent outbreaks have sort of confirmed that it's a real disease with organic origins. So it's not something that's necessarily in the mind, as it were. There's physical changes in biology in various organ systems that can contribute to this. And in 1995, the Center for Disease Control in the USA put it on as a priority 1 listing as a new and re-emerging infectious disease. And right up to 2019 now, we're interested in the possibility that it could be a virus that invades both nervous cells and tissues in the brain, and that this virus may actually gain entry to the body through the gut. So this sort of strengthens the gut link in ME. If I go to the next slide. So the evidence that there may be a gut origin for ME CFS is listed on this slide. So I think most of you can appreciate, and many of you probably have had GI symptoms of some shape or form, something like irritable bowel syndrome, which seems to be very common among ME sufferers. There's also a high incidence of enteric viruses. These are viruses that live in the gut or gain entry to the body through the gut. And associated with this is a leaky gut. So this is a loosening of the cell interactions in the gut, which provide a physical barrier that stop things that we eat and things that are in the food getting into the body. But as these junctions between cells leak or become leaky, then things like viruses and bacteria, bacterial toxins can get into the bloodstream. And one of the consequences of this is there's an increased sensitivity to food allergies, for example. And not surprisingly, it's also associated with this are changes in the makeup and function of gut microbes. And this is characterized by a decrease in diversity. So that's a reduction in the numbers and types of microbes that are in the gut. And this goes hand in glove with an increase in bacteria that we know are associated with harmful effects in the body, and a decrease in beneficial bacteria that are important for maintaining health. So this has led to the idea that maybe as a treatment for ME, we could be looking at how we can manipulate or change populations that got microbes to improve the disease and symptoms. So just to give you an overview of the microbiome, there are two terms that I'll keep using, so it's important I mention them now. The microbiota, which is a scientific term that describes all the microbes that are living in a particular environment. So in this case, the gut. So all the bacteria, the viruses, the fungi, the protozoa, they all make up the microbiota. But if we also consider their genetic material and all the genes that all these microorganisms encode, then we call that a collection of a microbiome. So that's including the genetic material that all these organisms in the microbiota have. So from several studies over the past 7 or maybe 10 years, we've been very good at counting, collecting, and identifying what's in the gut and in the human microbiome. So it contains over 100 trillion microbes, and the diagram on the right shows that viruses are the most dominant constituent of microbiomes, followed by bacteria, and then small numbers of fungi, which is things like yeast, archaea, and protozoa. And in total, there are probably about 1.3 to 2 times as more microbes in our body than we have cells in our body. And if you then include all the genetic material they have about 200 times more genes than we do in our own genomes. So there's an interesting thought there as to who's sort of living off who. And are we just a convenient home for most of these microbes? By far the largest microbiome we have is in our gut, where, as I've said, viruses predominate. Regarding the bacteria, there could be as many as 10,000 different species or types, which gives it that diversity I talked about earlier. And importantly, each of us has a unique composition or a microbiota. And this, if you like, is our microbial fingerprint. Just like the fingerprint on our hands, we have a microbial fingerprint in that we each have a unique microbiota. And this extends even to identical twins. Identical twins can be distinguished based on their microbial fingerprint and their own microbiota. However, within that vast complexity, all of us share a common core of about 50 different types of bacteria. So we all have this universal core. And this universal core is present in all of us because it plays an essential role in helping us digest our food. And it also is an important source of vitamins that we can't get from any other source, including a lot from our diet. Now, the microbiota is not only just there to help digest and process the food we eat, it also plays an important role in stimulating and maintaining the health of our immune system. And increasingly now, we've come to appreciate that it plays an important role in preserving mental health, cognition, in particular, and this is through the gut-brain axis. So the gut and the brain are directly connected via major nerves, the vagus nerve, but also by the blood supply. So what happens in your gut can very much influence how you behave, how you think, and how healthy your mental health is. So
Professor Carding, could you just
yes.
briefly, I've got a question, what are archaea?
So archaea are called ancient bacteria. And they are very interesting because they share properties or features of our own cells as well as bacterial cells. So some people believe they may actually be one of our sort of early ancestors way, way back that allowed us to develop as a multicellular organism. So they're found in very extreme environments. So most of them are found in things like deep sea vents or in volcano craters. So they're well adapted to live in very extreme conditions. So they've been called extremophiles because of that. But we also know only in the last few years that they also live in the gut, and they perform an important function in food digestion. So archaea, if you like, are ancient bacteria. So the cartoon I'm showing now is just a depiction of the importance of a balanced, highly diverse microbiome for preserving health. And if this balance or the type of microbes changes through various insults, then this can lead to the disruption of the normal functioning, allowing harmful microbes to grow or bloom or increase in their number in the gut. And this can then lead to what we call dysbiosis, or changes in the microbes, which is a feature of many diseases. And so what are the factors then that shape our microbiome? And basically it comes down to who we are and how we live. So our lifestyle and how we live is very important, because we're continually acquiring microbes from our environment, as the cartoon in the middle shows. So we can get them from a variety of sources, including our children, our pets, through the environment, things we touch, things we get exposed to. Diet is one of the things that can change the microbiota as well, depending on what we eat, when we eat, the type of foods we eat. Medications, in particular antibiotics, can have a very profound and long-lasting negative effect on our gut microbiome, as can gut infections. So if any of you have ever suffered from food poisoning, you know it can take a long time to fully recover from that. And some people believe that it can take several years for our gut microbiome to recover after a bad infection, and the same for drugs. So drugs and infections have the biggest and long-lasting effects on our gut microbiome. Everything else is a lot more subtle and generally more short-lived. Interestingly, as we age, our gut microbiome changes as well. So we start off, we're born sterile. We acquire microbes from our immediate environment, where we're born, how we're nurtured. These all influence which type of microbes colonize our body and gut. And then as we get into old age, we start to see a decline in diversity of our microbiome. We lose beneficial microbes. And this goes hand in glove with lots of age-associated conditions, cardiovascular disease, cancer, and increased susceptibility to infections, particularly viruses, and poor responses to vaccines. This is all in association with the age changes of your gut microbiome. So as I said, we now think that dysbiosis, that is changes in our gut micropopulations, are linked to lots of diseases. This slide just illustrates the possibility that it could be as virtually all diseases, greater than 90%. And in red there, I've highlighted ME/CFS. There is increasing evidence for a link or an association with this in ME. We can see a lot of diseases are linked to this. And this is really where we started off our ME research about 5 years ago, asking the question, is the gut the site of origin of ME? And so with the help of the Invest in ME charity, who've provided funds to support 3 PhD students, along with the University of East Anglia, we've begun projects looking at the microbiome, the gut microbiome in ME patients. This is the Quadram Institute. This is where I spend most of my time. It was open, it's a brand new building. It was only opened in April of last year. So hence, this photo was taken just before we moved in. But that photo is probably pretty close to what it's like now, actually, because we're all basically in lockdown and nobody's currently in the building. So the Quadram Institute, its remit is to understand how food and gut microbes promote health and prevent disease, with a particular emphasis on diet and age-associated conditions. So it has about 200 scientists in there. In the basement, we have the NHS, the Norfolk and Norwich University Hospital endoscopy facility. So there are patients coming into the building a lot, so we're directly connected to the hospital that way. And the BBSRC is the research council that provides money to fund most of our activity in there. And so in thinking of our ME study, we wanted to focus on patients with the clearest diagnosis and the most severe symptoms in order to be sure that the patients were actually had all the symptoms that were consistent with being ME. I'm sure a lot of you are aware that there's a lot of classifications that can be used, and these don't necessarily agree, particularly for patients that have mild to moderate severity. Depending on which classification is used for diagnosis, so patients can be diagnosed having mild or moderate severity, or maybe not even ME. So we made the deliberate decision to focus on patients with severe or very severe conditions. So that is individuals that are housebound or bedbound. And it's interesting, in sort of preparing to undertake the research project, we actually looked at how often severely affected ME patients have been included in research projects. And this slide just basically summarizes the fact that they are very rarely included in any ME studies, despite the high prevalence of very severe ME among ME in general. It may be as many as 25% of all patients have severe ME. So there are really too few studies that have been undertaken to address the disease in severely affected patients, which is what we focused on. So I'll just talk about one of the studies that's ongoing at the moment. This is looking at the virome. So we're particularly interested in the gut, maybe a source of viruses that can trigger or cause the disease. So this slide just summarizes what we know about gut viruses, and there are 4 different types of viruses. There are what we call eukaryotic viruses, which most of you, I'm sure, will be familiar with. So these are things like rotavirus, enteroviruses, herpes virus, cytomegalovirus. These are eukaryotic viruses, viruses that infect the cells in our body. And then there are viruses that can infect bacteria, and we call these bacteriophages. So these are viruses that infect bacteria. And the phages are, in fact, the most abundant viruses in the gut. And then there are smaller numbers of viruses that infect these ancient bacteria called archaea. And then we have things called virus-derived genetic elements, where actually maybe just be viral genes and not intact viruses. So the prokaryotes, so the bacteriophages that I've talked about, they are in fact the most abundant living entity on Earth. They're present in vast numbers in the ocean, and they can contribute significantly to carbon emissions each year, which is often not appreciated. They're in the soil, they're in sediments, both in the ocean and on land, and also there are a huge number in the gut. So there I put a number of 10 to the power of 24, which is a septillion or quadrillion, which is just an absolutely massive number. I was trying to think of how I could sort of give you an idea of what that translates to, and somebody said, well, that's possibly, when you look out at night and all the stars you can see, it's probably even more than that, maybe all the stars that are possibly present. It's a huge, huge number. And we're continually acquiring new phages through the things we eat, the drink, through breathing, etc. We're always acquiring phages. We can end up in the gut. And not surprisingly, we secrete a large number of viruses as well every day through our stool samples. So you can see it's quite a dynamic mixture of viruses in the gut. Some are coming in, some stay, some leave. And so the questions we're asking regarding gut viruses and ME is, what viruses are there? Is the virome, or the viruses that are in severe ME patients, are they different from those with less severe disease? In other words, is there something unique about gut viruses in severe patients that distinguishes them from patients with less severe ME? And we're particularly interested in the viruses that might kill bacteria, because it's this interaction of viruses with the bacteria that probably accounts for the dysbiosis that I've described before, that's associated with about 90% of all human diseases. So it might not in fact be the bacterial change in bacterial populations directly that trigger disease, but the viruses that cause the change in bacteria that are important. So we need to be focusing more on the viruses that can kill bacteria. And so this is a summary of sort of how far we've got so far. So we have looked at bacterial populations, and we can see that severe ME patients have a different disruption in their gut microbes and bacteria than less severely affected. So we may actually have a signature of severe ME, distinguishing them from mild to moderately affected patients. We've also seen that gut virus populations in severe ME patients are different to those of healthy individuals that live in the same households. This could be parents or carers. And this has been described before in patients with inflammatory bowel disease and diabetes. So gut viruses may actually be more important than we've come to appreciate in terms of causing some of the symptoms of severe ME. And one thing we're looking at in particular is, because of this disruptions in gut micropopulations in patients, does this cause a disruption in the immune system such that the immune system now starts to attack their own the gut microbes in the body, similar to what's been described for inflammatory bowel disease? So we have a disrupted immune system in ME. So is that immune response in ME patients targeted or focused on reacting with gut microbes, which would explain a lot of the disturbances in various organ systems that are seen in ME patients. So the current investigations, things we're working on currently, are down there at the bottom. So we're very interested in the interaction of viruses with gut bacteria, and if viruses are a cause of ME. And as a result of this, do ME patients generate immune responses that react to their own microbes, like an autoimmune response? So that's what we're currently investigating. I just want to mention 3 new initiatives that are about to start. One is a GP ME fellowship scheme. Second one is a new PhD student that we've developed through the collaboration with the Swedish ME Society. And finally, a bacteria therapy or clinical trial in ME patients. So first of all, with one of the local NHS providers in our region, the Norfolk and Waveney, we've developed a scheme to basically educate GPs about ME and to inform them, get them involved in research projects so they can become ME champions in their GP practice, but also educate and train other GPs in this part of the country about more awareness about what ME is and the types of research that patients should be made aware of, potentially could contribute to, get involved in, but also the latest findings from research and new studies that will hopefully lead to interventions. So we're targeting newly qualified GPs, and the GP is based in a GP practice in Norfolk. They'll, for a period of 1 year initially. During this 1 year, they will spend at least 1 day of every week at the Quadram Institute, getting directly involved in our ME studies. And what we're hoping to do is to get this GP to go out with us when we go and talk to and interact with our severely affected ME patients, because severely affected ME patients rarely, if ever, I think, see a GP. I think very few of them make house calls now. So in this way, we can also get GPs direct exposure to severely affected patients. And the ultimate objective is to contribute to ME/CFS research, improving patient outcomes, and for that fellow to go on and promote ME and more interest in GPs to get interested involved in ME and ME research. So that's the fellowship scheme. This is the joint PhD studentship with the University of East Anglia, Invest in ME, and the Swedish ME Society. So it's a 3-year studentship. The students started in October, and the the purpose of the project is to look at eukaryotic viruses now. So that's the viruses that infect our cells in the gut, to see if there's any eukaryotic virus signature in ME patients. So are there viruses in the gut that can infect cells in our body that could cause ME? And so the last initiative is the therapy and treatment. And this is really from the perspective of, can we restore a healthy gut microbiota or microbiome in ME patients? And so I put this slide up here to sort of give you an idea of the sort of general approach we can take to this. And I call this lawn care. So at the top of the slide, we have a flourishing gut ecosystem. Now, this can be devastated by antibiotics, so allowing weeds to appear. So this would be analogous to treatment with antibiotics and then the emergence of harmful bacteria or the weeds in the lawn. So how can we address this? There are three ways. One is we can use probiotics. So this is live beneficial microbes. So the analogy here for lawn care is seed. We can try to reseed parts of the damaged or affected lawn. The other approach is we can try and feed the beneficial bacteria with prebiotics. And the analogy here is turf food. So feeding with prebiotics to promote the re-establishment of healthy microbes. But if the damage and extent is large, then we may actually have to think about replacing the whole lawn. And the analogy here, so replacing the whole lawn, which would mean replacing the whole gut microbiota. So depending on the extent of damage and the individual, we could use all three approaches. Now, probiotics have been used for quite a while in ME, but a 2018 review of all the studies carried out up to that point said there was limited evidence for probiotics being effective in ME patients. So what about the bacteria therapy and the lawn replacement? So what we have here is something called microbiota replacement therapy, or fecal microbiota transplant, FMT. And some of you may well have heard of this because it's been in the news quite a lot over the past couple of years as a wonder cure for certain types of diseases. So you may think this is a relatively new treatment therapy, but in fact it's not. It's very old. It goes back to the 4th century. It's still part of Chinese medicine. In the 4th century, it was used as a human fecal suspension taken orally to cure diarrhea and food poisoning. Subsequent to that, it's been used to treat a whole array of other conditions using something called yellow dragon soup. I'm sure you can imagine what that is. And then more recently, during the Second World War, German soldiers in North Africa used something called camel poop soup. And what they did was they observed that Bedouin tribesmen would often follow their camels collecting the camel poop, and they would then use this medicinally to protect them against dysentery. So the German soldiers sort of mimicked this, and then eventually I think German scientists identified what the active ingredient in the camel poop was, a particular type of bacteria, and that this has been now used and is still being used as a form of treatment. And what I've shown there is Bactisubtil, which can be bought in Germany, as a treatment for a variety of conditions, and it is literally originates from camel poop. First experimentation in humans was around 1957 by an individual called Stanley Fecal, who fed surgical patients their own encapsulated feces postoperatively. This was shown to be very effective at suppressing or preventing post-infections, post-surgery infections. And then a little later, a doctor called Ben Eisman used it to cure 4 patients that were near death from a form of colitis, and that was using fecal enemas. And then more recently, Thomas Borody, a clinician in Australia, has used FMT to treat a variety of conditions, including ulcerative colitis, as shown here. but to date, I think he's done more than 10,000 FMTs for a whole range of disorders. Experiments in animals showed that there are some safety considerations that need to be considered when deciding on donors and recipient. And that was through a study that was carried out in mice, showing if you took feces from an obese mouse and gave it to a thin mouse, the mouse would become obese. So there are some issues there about proper screening. But then I think the real breakthrough in terms of clinical medicine came in 2013 when a very nasty infection that's usually acquired in hospitals called Clostridium difficile was cured by FMT. The cure rate was 90% or more from a single treatment, and patients who were bedbound or going to the toilet 7 or 8 times a day within 2 to 3 days were up on their feet and their bowel movements had returned to near normal, and the vast majority of them remained disease-free several years. So this has proven so successful that it's now part of the NHS, and it's becoming a frontline, first-line option to treat patients with very difficult to treat C. difficile infection. So in terms of what else the FMT could be used for, the variety of conditions that come to mind: metabolic syndrome and diabetes, cardiovascular disease, cancer, dementia, and possibly ME. And so this is again referring to Thomas Borody, who I've mentioned at the Centre for Digestive Disease in Sydney. And in about 2014, I think it was, 2000, he reported a study where he'd been using FMT in ME patients, the vast majority of which had irritable bowel syndrome, and they had alterations in their gut microbiome, so dysbiosis. He treated 60 patients, 52 of which had IBS, and his FMT was not a raw stool sample, but a mixture of microbes that he'd isolated from the stool, 13 of them, and this was given through transcolonic infusion or a rectal infusion. Of the 60 patients, 42 responded, as defined by improved sleep, improvement in fatigue and lethargy, and 58% were still symptom-free at 15 to 20 years following up on this. And interestingly, the GI symptoms, the irritable bowel syndrome, was no longer present in about 88% of the individuals. So these were individual cases, small numbers, but I think you'll agree, very encouraging, which has led us then to basically further or more convincingly demonstrate the possibility of using FMT by undertaking a clinical trial. And so this is the trial that we are hoping to start very soon. It's the Restore Me trial. So it's a phase 2 clinical trial designed to look for efficacy. This is the question the trial is designed to address. Can FMT restore a healthy microbiome in ME patients and improve their physical and, importantly, mental health? So this is a snapshot of the design of the study. It will be carried out at the Quadram Institute, so it's a single site. It will be a double-blinded, placebo-controlled study. There will be 160 patients in all. The patients will be recruited through the East Coast Community Healthcare, which provides ME CFS services to patients in Norfolk and Suffolk. So they have over 1,000 patients registered with them. We'll divide the 160 into 2 groups. 80 will receive a treatment, 80 will receive a placebo, which in essence will be their own sample given back to them. This will be in the endoscopy unit, which is part of the Quadram Institute. The donors for this come from a very select and carefully screened donor bank, and it's the same donors that we use in the NHS in the Norfolk and Norwich Hospital to treat C. difficile patients, infected patients. The distinctive features of this trial are that it will be the first clinical trial to assess efficacy of FMT in ME, and we will be using objective outcome measures. So it won't be questionnaire based. We'll be using objective measure to assess physical activity and cognitive function. So this is the study team and the design in terms of the timings and everything. You'll see it's a large number of people involved in the study that covers a large number of medical disciplines as well as research disciplines, gastroenterology, clinical microbiology, clinical trials people with an interest in aging, dementia, and then bioinformaticians, clinical trial specialists. It's a large group of people. And the data that we'll collect, we will have questionnaires, but the questionnaires are designed to assess symptomology, but also cognitive function. Individuals in the trial will wear an activity monitor called an actimeter that will feed back to the study team measurements of activity and types of activity. And then we'll be taking fecal samples and blood samples to assess engraftment of and stability of the transplanted microbiota, and the blood sample to assess biomarkers that are associated with disease activity in the patients. So these are the outcome measures. What we're hoping we'll be able to say at the end of it all is after 3 and 6 months post FMT, there will be an overall improvement in physical activity as detected by the wrist-worn actimeter you can see in the top right corner. And the graph there just shows you the types of activity that we can measure. 5-minute, 1-minute, hourly intervals, 24 hours a day. And changing cognitive function that we'll assess through various questionnaires and app-based systems, looking at memory, executive function, cognition in particular. Then the secondary endpoints will be, how safe is it? How acceptable are patients to the therapy? Are there any improvements in any pre-existing symptoms, particularly those that affect the gut, so irritable bowel syndrome, for example? And of course, we have to report any patients that have experienced any adverse effects, and that's a measure of the safety and tolerability of the procedure. So this was the timeline that we had originally. We've had to redesign and reconfigure a laboratory within the Quadram Institute that would meet strict requirements by the medical regulatory body, the MHRA, because the MHRA have said that human stool needs to be treated like a medicine or a drug. So it has to be prepared under very stringent, sterile conditions, and these have to be consistent. So we have to comply with that. We have to apply for a license in able to use FMT for this trial. We also obviously need to get ethical approval for this as well from regulatory bodies. And so we're hoping that all of this would be well underway by now, and we'll be ready to start sometime in July and August. But then, of course, COVID-19 has come along, and this has pushed everything back because the government offices and departments that were helped with the MHRA and the ethics application review have all been diverted to COVID-19 related activities. So we have to wait for the current crisis to abate somewhat before we can know whether and when we'll be able to restart. But we're still hopeful that some of this at least will be starting this year, and maybe we can actually start the trial near the end of the year or the beginning of next year. In the meantime, working with the Invest in ME research charity, we've been raising money, and we now have all the money we need to undertake the trials. Everything is in place. We just need to basically get back into the building to start the process. We are involved in another FMT trial, which is underway in Norway, called the Comeback trial, that some of you may have heard of. So we are involved in looking at the microbiome in these Norwegian ME patients before and after they've had the FMT. The study design is different to Restore Me in a couple of important ways. The first one is how the FMT is delivered. In our study, it's through a soft rubber tube, which is inserted through the nose directly into the small intestine. It's called a nasojejunal tube. The procedure is painless, there's mild discomfort, and it takes about 10 minutes. In the Norwegian study, it's delivered by an enema bag, which involves putting patients on tilt tables to ensure that gravity can have an effect to ensure that at least some of the contents of the bag actually make their way up into the lower regions of the gut. And their methods of assessment, their outcome measures, are not objective, they're subjective. They're based on questionnaires, and in particular the Child Fatigue Scale. So there are no objective measures there, but some of the outcomes, particularly the secondary ones, are overlapping with ours. So because this study is already underway, we have the benefit of learning from the experiences of Norwegian clinicians, and we still have time to find adjustments to our own protocol to learn from what the Norwegians are doing. So this is my last slide. Just thank you all for listening, and this is to acknowledge the people and the funders that have helped us with this. There are a couple of Twitter accounts there that you can use to follow us as and when we make progress on this. So I'm now very happy to open for questions. If I shut that down.
Brilliant. Thank you so much for a very, very clear presentation of the whole field of work that you're involved with, and really appreciate that. I think for me personally, there were some things that really stood out. One is massively welcome the focus on working with patients with severe symptoms, with severe ME. That's something that's always kind of really stood out about how variable some of the other studies can be, and that seems like a really critical thing to do, so that's great. And also hugely welcome using objective measures. Again, that's something that is so absent in so many studies. And also the involvement of GPs who clearly need a much greater level of understanding and access to up-to-date research to inform their practice. So those things are kind of especially welcome over and above the content of the research itself. So it's also very interesting to me, the prevalence of viruses in the microbiome, which I always associated much more with bacterial matters. We've got quite a few questions, some of which you possibly feel like you've covered, but I just want to double check whether you want to add anything. Sure. We had a query about the work of Vincent Lombardi and de Moyer about whether if you do manage to achieve changes in the microbiome system that are beneficial, whether they really can be long lasting. You have touched on this a couple of times, but I wonder whether there was anything else you wanted to add to that.
Yes, so I know there's been probably the most frequent attempt to try and change gut microbes in any human condition is through probiotics. So this is using either single or mixtures of purified bacteria that are then administered usually daily for long periods of time in the hope that they will establish themselves in the gut and give a long-lasting effect. Now you can buy things like Yakult, Actimel, they're all probiotic formulations. But there are issues with all of them. We don't know how many live bacteria in a yogurt pot, for example. How long has it been on the shelf? How long are they still alive by the time you drink them? And can they actually survive transit through the GI tract, through the acid in the stomach, to make an effect in the gut? If you think about it, what you're doing is you're adding one species to what may be 10,000 species in your gut. So it's literally a drop in the ocean, if you like. So the only way that you can overcome that is taking bigger and bigger doses, which becomes a problem in itself, but having to take it for a very long period of time to sustain it. Of course, from a commercial perspective, that's beneficial to them because they're making more money from you as you buy more and more of these yogurt pots. So the other approach, and cheaper approach, is prebiotics, and that is feeding your gut microbes with what it is they need to stay alive and healthy. And that is basically fruit and vegetables, plant-based material. That's what they're designed to break down and feed off in order to extract the nutrients from that we need for our own body. Because we can't metabolize complex plant material. We just don't have the enzymes in our genome to break it down. So we need our gut microbes to do that for us. So prebiotics keeps them healthy. And, you know, that comes back to the government's 5-a-day advice. You know, 5 servings fruit and vegetables should be enough to keep your gut microbes healthy. Some people say it should be 10 servings a day, but 5, if you can achieve 5, that's pretty good. And then the other approach is the transplantation approach, to try and push all the bad microbes out of your gut in one go and then replace it with a healthy population. So I think prebiotics and FMT are probably more effective in the long run for keeping a healthy gut microbe population.
Okay, thank you. There were quite a few questions from Robert Saunders, from Glenys, from sorry, a couple of other names as well, which particularly asked about pre and probiotics and off the shelf, off the counter, over the counter things that we could buy, and whether they could be useful. There's another kind of follow-on query from that, which is, could any of the kind of bacteria or the microbiome systems introduced through the trial be overridden again by sugar-loving bacteria or by the diet of the participants? How would that affect your outcomes?
Yes, that's interesting. Actually, in the hospital, when we're giving FMT to patients with C. difficile infection, the only thing we tell them to avoid for the first week after the procedure is medication, drugs, particularly antibiotics. So we don't tell them to change anything or make anything different in lifestyle and behavior. Keep eating, keep what you were doing before. Although there is advice provided from dietitians to ensure that the diet you have post-FMT is balanced and healthy. So that advice is given. But really, we don't ask people to stick to a strict diet to preserve their gut microbiome.
Interesting. Okay.
I should say, I forgot to say that the best food is fermented food, because they're called superfoods. And that is because you get probiotics and prebiotics in one go. So you've got the bacteria that have produced the fermentation effect on the food. And of course, you've got the plant-based material in the food as well. So fermented foods are superfoods. So if you really want to keep your gut healthy, gut microbiome healthy, eat lots of kefir.
Okay.
And fermented food.
Very interesting. If any people watching want to send in any more detailed questions then, or if you don't feel that Simon Carding has addressed those issues enough, then please send us more questions. Thank you.
I'd just also like to say we could open up the floor and people are welcome. If they post in the chat group, I can unmute them and they can ask questions directly of Simon Carding if they would like to. So give me a heads up. Thanks.
Thank you. Okay. And just following on from that briefly then, are probiotics still helpful if there are harmful bacteria in the microbiome?
So in a clinical setting, probiotics have been shown to be beneficial. I mean, bladder cancer is one example, traveler's diarrhea, infant diarrhea. But the types of probiotics given by doctors and hospitals is very different to what you can buy in supermarkets or health food. So generally they are super strength, so high numbers of bacteria, and they may contain 6, 7, or 8 different types of bacteria. So in a clinical setting with the right mixtures and right dose, they can have very beneficial effects. And you would actually need a prescription for some of those.
Okay. And a query from Michelle. Is it possible for people to get their own microbiome tested, and would that produce any useful information that people could do on their own, or does this require involvement in a study?
There are a couple of, there are quite a few organizations, particularly in America, that will profile your gut microbiome and tell you how healthy you are and what things to avoid and what things to eat more of or whatever. So American Gut is one program, a system, so you can send it off, and I don't know how much it is, but it's not cheap. But the important thing to bear in mind is that your microbiome shifts very quickly in response to what you eat and what you get exposed to. For example, if you significantly change your diet, then your microbiome will adapt to that, or start to adapt to that, within 24 hours. So it will shift and change and differ because it's responding to what you eat. It needs to adapt. So, and also, we also know that different populations of microbes expand or become dormant at different times of the day. And again, responding to what we eat and where we are, if we're outdoors or indoors and what we're exposed to, it can shift and drift. So when you take your sample, it becomes very important if you are going to have this done. And so ideally what you would want to do to get a true and accurate picture is to give different samples from different times of the day or different weeks to get an accurate overall picture rather than a snapshot, one sample, which may or may not accurately reflect what types of microbes are in your gut. So I would say, don't waste your money on them.
Certainly the ones I've seen have come up with some very, very generalized advice at the end. I've not had mine done, but yeah, which didn't seem that
and of course,
helpful, really.
The other thing to bear in mind is it's an incomplete picture because these tests will only be looking at the bacterial component. So, they're not able to look at all the other microbes there, which could just be as equally important for maintaining a healthy microbiome. So you're also getting a very narrow picture of what's in your gut at that time.
Okay. And a further question then, which builds on that quite well, from Anil van der Zee. So welcome to this session. What about antivirals treating the virome?
Yes, so in comparison to antibiotics, we don't really have effective antivirals yet. We have a few, but they're very small in number. But the other thing is that our understanding of viruses in and on the body and in the environment is very limited compared to what we know about bacteria. And that's because we've not been very good at isolating pure viruses from humans or samples or patients, whatever, to give us a reference database by which we can then identify others in other samples. So one estimate has that we may know or be able to identify less than 20% of all the viruses in any virome, be it the gut, be it the soil, oceans, whatever, because we just don't have enough reference viruses. We don't know enough about them. So we have something called a viral dark matter, which is probably 80% of the virus population living in the gut. We've no idea what they are. So it's a huge undertaking. So in terms of thinking of therapies, it's very difficult if you don't know what it is you're trying to target. Okay. So antivirals would be good for some viruses where they've been shown to be very beneficial, but it's a bit of very much hit and miss, I think, if you don't know what the actual virus is. And viruses are very good at hiding in cells, hiding in the body and hiding in bacteria. So they're very difficult to identify for that reason as well.
Okay. You made mention during the talk about how vaccines can potentially change the constitution of the microbiome. Are there any studies, this is a question from earlier on a bit ago, are there any studies that look at before and after impact of vaccines?
I'm aware of one study that's underway in Denmark looking at the HPV vaccine. Because I think there have been some cases of ME or ME-like symptoms in young women that have been given the cervical cancer vaccine. That study, I think, is being written up now. That's probably the one I'm most familiar with. I mean, most viruses are administered via injection. So the gut wouldn't necessarily get a large exposure because the vaccine would disseminate through the body. And that's actually a problem for most vaccines, because if we're thinking about COVID-19 infection, for example, or influenza infection, we want vaccines that give us protection in the lungs. Because if we can stop the virus in the lungs, then we don't get all the systemic effects of the virus infection. We don't end up on ventilators, we're not killing so many people. 95% of the vaccines we have are injectable vaccines, so they're not particularly good at providing frontline protection in the gut and the lungs. So what we need are more vaccines that target those, that are delivered orally or delivered intranasally. And the best example for an effective oral vaccine is a polio vaccine, which interestingly didn't cause any significant changes in the microbiome as far as I know. So, oral vaccines can both be very effective and also have no adverse effects on the microbiome.
Okay, thank you. There's a change of tack, question from Robert Saunders and from, I can't read this, it's just disappeared off my screen, about models for ME and fibromyalgia with mice or in mice, and also whether we can transfer a model of the microbiome into mice.
Very interesting question. So there are no useful animal models of ME. There is no animal model that can faithfully replicate the key symptomology that patients have. So that's a major problem, because for most other diseases, there are animal models in which you can understand and look at the mechanism of the disease, but also you can test drugs and interventions in. So that's a real issue we have for ME research. There are no useful, as yet, animal models. Now, the question about transferring human samples into mice to make a humanized mouse, for example, which has been done to look at the gut-brain axis and how gut microbes might influence the development of Parkinson's disease. So we've shown if we can take a mouse that is genetically engineered to develop a Parkinson's-like symptom, if you take a stool sample from a severely affected Parkinson's patient and put it into that mouse, it accelerates the progression of the symptoms. So that's the strongest evidence we have for a link between gut microbes and causing a type of dementia. Now, could we do that for ME? It's possible. I'm aware of a couple of attempts to do that which have failed. So that could be because the animal model's wrong. Maybe they shouldn't be using mice, you should be using some other model. Or you've sourced the stool sample from the wrong sort of patient with the wrong symptomology. Lots of questions there. But I think potentially that is something that could be investigated further, is trying to humanize an animal with samples from an ME patient to try and replicate some of the symptoms that we can then at least start to begin to investigate the causes and treatments. So I think that's the desperate need that the research community has. It's a good model system.
Okay.
Because ethically, of course, we can't do these experiments in humans.
Indeed, indeed. I've got a series of general questions about diet, but a very specific one first. Is there any evidence for Borrelia burgdorferi in the gut? Sorry if I've not pronounced that correctly.
Borrelia, yes, but it's not what we call a commensal. So it could be a transient, so it's there, but then it doesn't stay, it gets pushed out, is lost. So again, depending on the situation, the conditions at which you take the sample to analyze, it could be there or it might not be there. It's certainly not one of the core members of the microbiome. So it could be similar to Helicobacter pylori, which is associated with ulcers and stomach cancer. So about 25% of the population have Helicobacter as part of their microbiota. Fortunately, it doesn't cause condition in the vast majority, but 75% of the population don't have it. So there are probably going to be other subgroups of individuals that have what we might think as a nasty, harmful microbe, but it's not causing the disease in that patient. It's not present in most people. So yes, it's entirely possible, but it's not a general commensal.
Okay. So we've got some broader questions about if a plant-based diet is good, what about dairy, what about cheese, and what about fermented cheeses like Roquefort is one of the examples given.
Whoa, yes. So I think you know, like it's all foods. A little bit of something is not bad for you, but too much maybe. And I think with cheese and dairy products, that is probably that fits into that. So there are things in dairy products which are good for us, are beneficial. So for example, there's an important probiotic bacteria that lives in the gut called lactobacillus or lactococcus that feeds off principally sugars that are present in dairy foods. So it's good to promote their growth, but too much can cause the growth of less beneficial microbes in the gut because of the other things in the products you're eating. So yes, I think it's like with most things in life, you know, a little bit is not going to harm you and could be beneficial, but too much could tip the balance towards you know, harmful effects. But fermented foods in general, I think, are more beneficial than just taking prebiotics or probiotics alone.
Okay, thank you very much. I think I've covered most of I haven't covered every question in great detail, but I think I've covered most of the themes. So I was going to ask as well, you partly covered this, which is like, what is the impact of COVID-19 on your research? So I think you've covered that. I was just wondering more broadly, and I suspect it's too early to say, but whether you're aware of COVID-19 opening up any new forms of potential research about the impact of viruses in the context of people who then become symptomatic with longer term post-viral syndrome and ultimately ME.
Yes, so there's some interesting studies which have identified COVID-19 in stool samples of infected patients. Mm-hmm. And that's a molecular detection-based system. So as yet, I don't think anybody's isolated intact infectious virus from stool samples of patients. But there is a thought that this could be another route of transmission, should there be any live virus in stool samples. We know that the virus can infect cells that line the gut, so it's possible it could be there. So this could be another route of transmission, the fecal-oral route. So just yesterday, I was part of a conference call with members of the European ME Research Group, which I co-chair with a Danish colleague. And this organization has come together to try and unify ME research across Europe. And the membership includes about 8 or 9 different countries in Europe. And so we're trying to coordinate research so we can all basically contribute to various projects in different countries using patients in different countries, etc. Mm. And one of the things we discussed yesterday was initiatives that we can take to track COVID-19 patients to see if any or how many later on develop post-viral fatigue and maybe ME. At the moment, it's an interesting question. Now, if we were able to detect ME in patients that recovered from COVID-19, then that certainly is a smoking gun. And again, would fit with the infectious origin, viral infection origin of ME. But it's just too early to tell. I mean, we have hospitals, we've got clinics in 5 European countries which have the ability to track and monitor patients after they've been discharged from hospital. So we are going to initially focus on those as a means of tracking and monitoring and looking for the occurrence of ME in the patients. I mean, that comes about just because we've got the organisation and the contacts to do it through the European ME research groups.
Okay, brilliant. There's another question coming about a low FODMAP diet. I think you've kind of partially covered that by particularly talking about fermented foods. So I don't know whether there is any more you'd like to add to what you've said. And the other question, I presume it's Robert Saunders, is it possible that there may be pathogenic microbes which are attached to the gut wall, which are not picked up in sequencing of stool samples? And is there any way to test this? So, you know, could we be missing anything?
Sure. So the first question is the jury's out on FODMAPs. I mean, I've seen persuasive evidence for and against, so I just don't think the studies have been enough or done well enough to be any conclusion about that. For sure, regarding the second question, when we take a stool sample, that is a surrogate of what we believe to be in the gut. But as the questioner was quite rightly said, if bugs attach very tightly to the gut wall, they're not likely to be in the stool sample, or if they are, in very, very small amounts, so they could be missed. The only way to sample them is through a biopsy, which I know for ME patients is not part of any diagnostic procedure. So that would be something that would have to be done as a research-only protocol, which involves ethics and approvals and health and safety, lots of things would have to be done to show that that would actually be beneficial. So yes, it's just through a biopsy, I'm afraid, a colonoscope.
Okay, thank you very much. Well, can I say a massive thank you to everybody who's joined us today. You'll find in the chat room we've posted a link to a feedback form, because we always welcome feedback on these talks that we host. So please head over to fill that in as well. So I've still got more messages coming in. I'm trying to read them as I speak. So right, oh great, Kerry. So fascinating ideas. Hoping next year to do my dissertation on something relating to ME and gut nutrition. So go, Kerry. Please do. That would be great.
Absolutely.
So a couple of particular thank yous. One is to Sian, who's in the other room here, who's been doing all the technical, making sure everything flows smoothly. Thank you to Isabel Hemmings, who's one of the trustees of the Sheffield ME Fibromyalgia Group, who made the initial contact with Professor Carding. So a massive thank you to Professor Carding, to yourself, for being willing to do this talk. And it's obviously unusual circumstances. We'd have loved to have had you in person. But in fact, we do, we are reaching people around the world as you speak. So whether you were in person or not in Sheffield, a number of people would have been online anyway.
Sheffield's one of my favourite places to visit.
Oh, me too. So
really
please come and see us again. Yes,
I'd love to.
Please come. You'll always be welcome. The talk has been fantastic. It's brilliantly pitched, and I've understood virtually all of it, and that's really good. And it's also given us some sort of really positive hopes and opportunities for the future. So I know your work is very much supported by Invest in ME Research. They've done an incredible job fundraising and will continue to fundraise. So
thank you very much.
So please everybody, if you feel able to make any contribution to that research, please do so on the Invest in ME research sites. And yeah, you had a lovely comment from Anil van der Zee, who's in the Netherlands, saying rockstar Simon. So that feels like a really nice ending from one of your fans over the water. Yeah. So yeah, so thank you very much for joining us today, and really appreciate